Hello, everyone. My name is Bo-Kai. I'm the final speaker of the year. Sorry. And okay, so I'm from the Department of Aquaculture, and I have to apologize. The only species I can aquaculture is river fish. I know a little bit about river fish. So my personal interest is self-organizing training. And today I want to show you a quick story about our recent study on river fish embryonic skin about the stochastic determination of ionocyte. And sorry. And so for the determination of ionocyte, which is a spatial type of cell, they responsible for ion regulation. So as we know, river fish are freshwater fish. So those cells are responsible for uptaking ions for the fish for development. And those cells are scattered on the epithelium, on the mostly on the yolk ball region, from the 12-hour to before the gill go out. And this process has been long-term been known as co-particle lateral inhibition. So the delta will inhibit, sending the inhibiting signal to each other. And as a result, after this competition, the winner will be selected. And they will form a typical scatter or salt and pepper pattern, like this one. And this can be either visualized by the changing line we met before in my lab. You consider it's the delta C reporter line, not reporter, delta C fusion Venus line. So, and or you can assume it on the computer. And but this is not the whole story. So if you this is just progenitor of ionocytes, and this has been published at least 5 subtypes ionocyte. And here I'm going to show you 3 of them. And this is a triple staining of one river fish embryo. And if you look carefully, they are kind of each scattered with each other. And I won't bother you with the ionocyte name. Here's HRC, NRC, and NCC, just 3 different types here. So the NCC is in situ, so it's a bit dark here. But you can see they're scattered with each other. So this gives us one like a general question: whether is this cells they all come from the same progenitor and they are determined by a stochastic, like say random fate determination process. But so far the best knowledge we have about this process is about 2 genes. It's FOXI3A and FOXI3B, and it's being proposed these 2 genes interaction with each other, and they will determine which subtype they are. But so far, it's not so clear how they're doing this. It's how these 2 genes can determine at least 5 types of cells. And so hopefully I can, but I guess I will going bring you more questions and answers. Okay, so before we try to study whether they are stochastic, we take a bit step back. We ask whether all those epithelium cells they are stochastic, or how they distribute on the skin. And we use really stupid and simple method. Let's a lipid cell mask. It will stand on a cell membrane. And we use another program published recently, so called Cell Pulse. It's an AI deep learning program, can help us to identify the cell boundaries. Quite useful. And then we get the coordination of the apical opening and do a simple mathematical test. It's called error function test. Have been proposed quite a long time ago by Ripley, so it's called Ripley K function. And this function is really simple, and the L function is a linear solution of this K. So it's and this mathematical is so simple that I can understand, I can perform. And so the result is easy. So it's basically X for each coordinate given a gradient increasing ring, and count how many other particles or spots in these circles. And the result is really simple. If this dot they scatter randomly, you will get a perfect straight line in the middle in here. And if your line is upper than this one, they are clustered. And your line, if down to here, they are regular, so they are from distributed in an even space. And in particular space, as a particular case, they can go like a regular in the locally and cluster in the globally or vice versa. And let's look at the epic opening first. So the epic opening, we can count them from the cell mask and soil project for this image quality. And so it's for simplified. So we label a cell boundary. And the epic opening is located in this we call tricellular junction. Tricellular junction can formed by the keratinocyte and they provide a small space for the ionocyte or mucous cell underneath, give them a small opening to face outside the body. And this opening is easy to like a control, can be open or become smaller, even sometimes sealed. So in principle, all the tricellular junction have one potential space for one this type of cell underneath. So we label all the location and do the run the error function test. So left-hand side is for the local. So each dot is on the coordinates a particular apical opening, and you can see they are all really regular, and especially when you look in the middle, they are in the this is a local comparison to the global regularity. So it's more yellow. It means it's more cluster because they are all located outside. It's by the nature of the mathematics, not about physical property. So because of the calculation, so we have to look in the middle. They are regular, and they also you look at the line. They are really down to the random guideline. So we know all the opening they kind of regular space. So it's simple to understand because you can see the keratinocyte is roughly similar in size, and the tricellular junction also have to be roughly have an even space on the surface skin. So then how about the ionocyte? So we do the same thing to these three types of ionocyte we have, and found out those ionocytes, they are kind of a cluster on the whole skin. And but if you're looking for these three in particular, because they have to adapt the total population, right? So if this line are overlap, it means that they are random. So roughly these three types are random in the ionocyte space. So if I hope I can explain better, but this is the red line here indicate all the coordination from the ionocyte, and each blue line is for each ionocyte subtype, and they are all overlap, so it means they are kind of random. Okay, so that means, in other words, in each particular position, they can be developed into either three of these types. Okay, and that means they are cellular fate is random. You can find this kind of analysis in like a retina, like in human, our pigment cell. We have in human we have three types, right? The red, blue, and the green color, and they are randomly distributed on retina. Okay, so then we want to understand what's going on during this determination process. So we use the background combinating to generate two transgenic lines, and we first synthesize two constructs. So one is FOXI3A, the FOXI3B. We label them with the red color and the green color for instance protein. And here's the two lines. So one is have both these construct, and they actually cause quite high copy number. So one FOXI3A has almost 100 copy number, another one has 60 FOXI3B copy number. But you can see the expression quite well. And another transgenic lines only have the FOXI3A, but in green color. So it also have almost 100 copy number transgene in this fish. So it gives us opportunity to track them. So of course we want to see the tracking, and this tracking is called a lot of surprising results, even for me. So I guess you can see roughly two things happen here. So those cells, they a little bit aggregate or cluster with each other, and which you won't see in the mature ionocyte. Another one is you can see particular one or two cells, they can moving really, really fast. So you can see that like moving really quick. We have no idea what's going on, and they kind of have their own direction to find another cluster. So this cluster are formed by partially from cell division. So once they divided and they don't separate, they just try to stick together, and also from aggregation. So so you can see the first time they have only two, but in the end they aggregate into 4 to 5. And I have no idea what's going on in this one. So, but this one we can check in single individual cell. So several type cell we found, they all have this, the green color is FOXI3b. So they have a fluctuation in expression. So some even a little bit like oscillation type, but some are not. So we still call them fluctuation at this moment. And so we want to solve which cell is which cell. So luckily we have another line. It's as a gift from Professor Huang Shenbing's lab long time ago. And this is a reporter line. So this report line is arbitrarily labeled, and they will label the whole cell, the cytosol part. So with these two lines combined together, we are able to give them identity when we check them. So for example, if you see the bigger one, bigger red fluorescent signal with a green nucleus inside, they are one type of this cell. And if they are solely a red signal, they are this type. And if they are green signal and wandering around by themselves, it's probably this type. And we can also give them, try to collect enough information for the expression. So for example, for this HRC cell type, they mostly stick together and they were divided during the determination. And for this one, they are usually single in the red and they are rare in this line. And for this cell type, they are in a single, they don't clash to each other a lot, and they have a strong green signal. So just roughly get this idea. But they all share one property is this jiggling around expression. They are not stable expressed in any particular gene. And so we want to try to understand how to get the same phenomena in a computer. And I skip some data. I don't want to bother you with too much like molecular data. So we identify a pathway like this one. So the delta is the same is the rate for lateral inhibition, and the A gene and B gene will interact with each other, and this form a really interesting loop. And the C will induce A, and the A will induce B, and the B will inhibit A, and the A will induce C. So we try to put this genetic network into computer, try to simulate the result. Unfortunately, I'm really bad in physics. That's the reason I study agriculture. And so, so far I only got the qualitatively result. Okay, those lateral inhibition selected progenitor cell, they will have several expression of the up and down of this A and the B gene because they inhibit each other. And they also, this feedback loop will give them into different time step, so make them kind of stochastic expression. And so for resolution, you have to, so it's another snapshot, another simulation. So they will first select some delta cell, the red one, and then the FOXI3A, the A gene, will start to express and induce B gene, and the B gene will turn off the A, and this A feedback will cause the C delta to prolong slightly longer and cause some cell into the oscillation type, some cell into stuck in different type. So in the end, the output can be different cell may have different combination in these two genes. So with different combination, it can be further developed into different cell types. And not many labs study the stochastic gene spread determination, but so far the example as we from the literature we found haven't have this simple way to describe this mechanism. Okay. So we in the end I want to thanks my lab and also the funding source. And also, sorry, give some chance to advertising. I'm currently editing for guest editor to editing a special issue in Frontier. If you have something related to patenting, please help us to survive for this special issue. I will do my best to help you. Okay, so so let's really quick. Thank you.